Hi everyone, I'm Pallavi N, PhD scholar, Department of Crop Physiology, University of Agricultural Sciences, Bangalore. I'm really very happy to be a part of Global Reach 2021, and I would like to welcome all of you for my presentation, which is on saline-tolerant endophytic fungi improve cellular tolerance to stress in a new host system, that is tomato. So, since plants are sessile in nature, they're continuously exposed to various stress conditions during its developmental stages. Among different abiotic stress conditions, salinity stress is the one of the most devastating, which is significantly affect growth and development and productivity of major crops. And it is estimated that at least 50% of the arable land worldwide will be salt affected by the year 2050, and this is expected to further increase due to global change. And in recent days, plant endophyte interaction is a growing interest and contributing a lot of significance to present era of agriculture. An enormous amount of research has been done to prove that these endophytes are one of the alternative approach to mitigate both biotic as well as abiotic stress conditions. Then what are these endophytes? These endophytes are non-pathogenic microbes. They live within the intracellular spaces of plants and feeds on the apoplastic nutrients without eliciting any defense responses. And according to the literature survey, they possesses many direct and indirect mechanisms. They ultimately bring about a series of beneficial activities. Direct mechanisms includes production of secondary metabolites, siderophore productions, phosphate solubilization, nitrogen fixing ability, and increase in phytohormones like IAA, GA, and auxins. And indirect mechanisms includes bioremediation or access. The endophytes act as a biocontrol agents. So based on the literature references from our own group, endophytes are used to manage salinity stress tolerance. And with this knowledge of endophytes, I collected few endophytes from the library, that is a School of Ecology and Conservation, UASB, and maintained them in a pure culture in slants for further use. And all these 10 endophytes were evaluated for their ability to impart salt stress on a PDA plates by asking a question that whether these endophytes are capable of growing under high salt concentration conditions, for which I took actively growing mycelial discs measuring zero 0.9 centimeter in a diameter were placed at the center of the PDA plates, and where PDA plates were amended with different concentrations of NaCl, and they were incubated for 5 days. And after incubation, fungal colony diameter was measured. And with the increasing concentrations of NaCl, the fungal growth were decreased. And based on the colony diameter, endophytes were selected. And among 10 endophytes, 4 endophytes showed increased fungal growth up to 2.5 molar concentrations of NaCl, and which means that these endophytes are capable of surviving under increased concentrations of NaCl and probably imparting salinity stress tolerance to host system. Since we wanted to pick a salt-sensitive genotype of tomato, therefore, we selected 4 genotypes of tomato, which are commonly grown, namely Arca vicus, Arca saurum, Arca megali, and Arca aba. They were surface sterilized and they kept for germination. Then uniformly germinated seeds were placed on a germination paper, which was added with different concentrations of NaCl, and they allowed to grow for 7 days. After 7th day of incubation, shoot length and root length were recorded, analyzed, and compared. Here I'm showing the seedling length of all the 4 genotypes, which is represented in graph, whereas x-axis indicates different concentrations of NaCl and y-axis indicates the seedling length. Here the blue bar indicates the shoot length and red bar indicates the root length. Here LC50 value was calculated using SPSS software and probit analysis. So LC50 value is nothing but a lethal concentration, which defines as like 50%, where the 50% growth is reduced. Here we estimated the LC50 value among all the 4 genotypes. Arca saurum was found to be susceptible or sensitive to salinity stress condition by recording significantly lesser seedling length. This study helped us to find out the sensitive genotype of tomato. So we have sensitive genotype, which was surface sterilized and kept for germination. And along with that, we have a saline tolerant endophyte, which was selected. and uniformly germinated seeds were incubated in a fungal suspension for 3 hours, and they were tested for their ability to impart salinity tolerance. After seventh day of incubation, seedling length were recorded, again analyzed and compared. Here I am showing all the four endophytes which showed increased mycelial growth even up to 2.5 molar concentration of NaCl. Here x-axis indicates different treatments, which includes control, endophyte, stress, and stress plus endophyte. And these are the images of seedlings which are inoculated with different uh, endophyte different endophytes after seventh day after germination. So from this we found out that endophytes can colonize into a new host system and thereby increasing the seedling length significantly. To test their colonization, seedlings were made into bits. They were surface sterilized and placed on a PDA plates and allowed to grow. Then they were compared with the mother culture by both morphologically as well as at molecular level. At molecular level by isolating genomic DNA and PCR amplification using ITS primers, and they were eluted and sequenced it. And sequences were compared, and then closest match was identified by constructing phylogeny using Megaten software. Apart from these, we also observed the morphology using SEM, that is scanning electron microscopy. Here I am showing the SFI, which performed significantly well under salinity stress condition, and this SEM image tells us that endophyte produces both macroconidia as well as microconidia. Macroconidia are fusiform, slightly curved, pointed at the tip, and having 3 to 4 septa. And these macroconidia are abundant but never in chains, whereas microconidia are smooth to rough walled with 0 to 1 septa. Then coming to conclusion, these selected endophytes can successfully colonize into a new host system and thereby imparting salinity stress tolerance in tomato. And SFI significantly improved seedling length under salinity stress condition compared to other organisms as well as to control. And our study indicated that stress tolerant endophytes can be used to improve salinity stress tolerance to tomato, which will have a practical relevance under field condition. At last, I would like to acknowledge or thank the World Bank funded project, that is Indian Council of Agricultural Research, Center for Advanced Agricultural Science and Technology on NGT for microbiome enabled seed priming, for providing me the financial support. And I would like to thank my chairman or supervisor, Dr. Nataraja Karba N, Professor, University of Agricultural Sciences, GKVK, Bengaluru, and who is also a co-principal investigator of this project, for guiding me in all the way. And I also would like to thank Dalhousie University for providing me this opportunity to present my part of research work. Thank you.